We've all been passionate on precision medicine, and we're going to take 5 different aspects of that. Massimo will start by talking about liquid biopsies, which he and I do absolutely routinely. Raju is going to discuss Raju is going to discuss discrepancies in the application of precision medicine across the country and perhaps around the world. Then after that, Min will follow, looking more at the GI aspect. Alyang will follow, who will look more at the brain aspect, and then Drew will come last. I don't know what you're going to talk about, Lou, but I think it's more immunology. But you can just sit there, don't worry. We'll bring you some coffee and buns. Anyway, this session is going to be quite unlike the last. The last was so brilliantly organized and went all the way through. This one, I want far more audience participation in this, back and forth. So Massimo is going to go first, talking about liquid biopsies. He will show 3 slides, and then I'm going to open it up just a little bit for questions, and then we'll do the next, and Raju, and the next, and the next, and the next, and we'll have a closing discussion at the end. I thank you all. Massimo.
Thank you, Brian. This is obviously a great audience, great topic. We are all passionate about liquid biopsy, but we are mostly passionate about helping our patients and improve their life and their survival. So I'm going to tell the story of the blood-based diagnostic, how we started, at least my journey, and where are we going toward which directions. The way we started listening to our patients, always. Patients with metastatic disease, newly diagnosed, comes to your clinic and ask, how long I have to live, because I was told 18 months, and this was 25 years ago, 18 months was probably within the range of what we expected. And I said, I don't know. Why I don't know? Because every book, every chapter that you talk about metastatic breast cancer is in 18 to 24 months. We must have a better way to understand the disease and the evolution of the disease outside the imaging. And so we started to work on circulating tumor cells. And at that time, there was only one technology. This technology allowed to enumerate the cells and enrichment based on epithelial characteristics. And we showed that, in fact, more than 5 CTCs were able to stratify patients with more aggressive disease versus not. What this means, more aggressive disease, we figured out later on that obviously these patients have not only higher tumor burden, but have a fast progression to metastatic disease in other organs. So these CTCs inform on the metastasis. Then we start to look at the actual images, and we identify clusters, extravesicles. So there is a universe of cells, or cancer cell elements that we had not studied completely, but eventually over the years we published on that. But what we also realized is that technology was not good enough to do all the other studies that we wanted to do. We wanted to stain the cells. We want to isolate the single cells, as was mentioned this morning. We want to grow cells, and that's what one of the NFRC projects that we are working on is isolate with microfluidic and try to grow organoids to test drugs in a metastatic setting. And during this, we work also with filters. We identify first the camels, that were the cells that we just mentioned, and we published that paper, and we show later on that the camels can also be seen even in patients that still don't have cancer. So maybe the immune response is faster compared to the cells in the bloodstream. In any way, we needed to be more practical because we need information about the genomic of the disease, the molecular evolution of the disease. So cell-free DNA became available, and we start to work on that and identify mutations, mutation of resistance, driver, even germline in the cell-free DNA. And of course, that was very important because it's now driving essentially drug development. I'm going to talk about drug development in a moment. So as I say, there is a lot that we can find in the blood. The 2 most important and established technology and modalities evaluating, identifying, and enumerating CTCs, looking at biomarker and CTC, and cell-free DNA, mostly NGS, looking at panels, obviously hotspot mutations. You can also looking at cell-free DNA as a measure of response, as a tumor burden, and this is very important, again, for drug development. One idea of drug development would be that using a cell-free DNA a baseline, you can probably look at the driver mutation and change your drug according to the mutation that you have. What is interesting is also the individual mutation of different variants, and now with the availability of different agents, SERD, for example, each one of them can have an effect on different variants. So we can really personalize the treatment. It can also predict the site of recurrence. So this is a graph just to show exactly where this cell-free DNA and CTC fit, essentially from the beginning all the way to the end. But in order for us to make the big move, that is the one we will talk in a minute, is the big move would be the screening. We need to first establish the sensitivity of the test. There's a lot of work that goes into the development of the technology. So we establish the technology allowing to identify the resistant mutations, for example. And this was in metastatic setting. Now it's incorporated in many clinical trials. It's just that we do this without stratifying patients, and sometimes we don't use the sequence. We have a molecular recurrence that can happen before the radiological recurrence. If you imagine this as a lag time, possibly this is an opportunity to improve the survival in the metastatic setting now that we have so many agents implementing before the imaging actually show the progression. The big area of the molecular residual disease after primary treatment. Again, our patients ask after primary surgery, what are we going to do now? Nothing. There is no recommendation. Well, we have now data showing that, in fact, we can identify, particularly in the triple negative disease and subtype, progression in the blood at least 6 months before this happens. So if this is the case, and we have more sensitive technology that the tumor informed, can we actually move and identify the disease later? We were talking about the survivors, the late recurrence. So this is obviously something that can be done. And speaking of the late recurrence, of course, there is not a number of studies that are looking at the use of some technology like the GRAIL, Galleri, in patients that have 3 years or more the disease already to identify new primary. And of course, you pick up also other tumors, in that case, directed toward the standard direction of selection of screening for other tumors. So the cell-free DNA has evolved, and now we can look at tumor burden monitoring in the neoadjuvant setting. Of course, there are plenty of studies that validate this. There is MRD detection. This is the next phase. Can you actually treat the MRD? And then with the MRD treatment, delay the early progression or maybe delay the recurrence. You need different technology for the two different questions so that we don't have to design study with 5,000 patients and give to everybody a CDK4/6 inhibitor, but we can reduce the cost by identifying the patient that I needed that. And then we look at the molecular recurrence for progression. So this is the ambitious next step, right? Once we have sensitive technology, we have validated the different setting, can we actually increase the screening? This is breast cancer month. Everybody's talking about mammogram. Still is important, it's the gold standard, but it's absolutely not enough. Sensitivity is very low. Young women, high risk of disease for genetic basis. Of course, access to mammogram. We need to do better. And I think we're trying to develop this technology in the early detection space. We just heard that we can also intervene with prevention in case of alteration that you can find in the blood that are not associated with alteration in the imaging. This is a counter-inception concept that we have different agents for that. So I just think about the many opportunities for women screening. How many women in an underserved region we can actually have a screening without having to go with a bus, with imaging. There are not enough breast radiologists, so we have a real issue. There are high-risk conditions. In New York, we have a large population of Jewish that gets breast cancer before they can be screened. So we have to do better for that population. Lobular cancer, you know, is not very sensitively detected by mammogram, and dense breast is always a major issue. So I think there is hope that liquid biopsy for breast cancer, possibly for other tumor types, can really be implementing from the first cell, probably not, but for the first event, the eventual will bring. And I am a proponent of using liquid biopsy more effectively for drug development. Thank you.
So Massimo, superb. Can I just ask, what do you do in terms of liquid biopsy? Do you do just cfDNA? Do you do CTCs? Do you do mRNA? Do you do any of the epigenetics?
So you definitely use cell-free DNA, almost a standard, particularly in hormone receptor positive patients. As you know, we have several targeted therapy approved in these patients. Alpelisib for PI3K mutations. Soon we'll have an AKT inhibitor. ESR mutants are very important. It can also drive the localization of the disease. CTC, as you know, is becoming an issue for insurance payment, but now is covered by insurance. I think CTC can stratify patients, as I mentioned, in terms of aggressive, less aggressive. You can do HER2 on the CTCs, and of course we have an HER2 for low HER2 expression, so I think this can be very helpful in some setting when it's difficult to repeat a biopsy. Obviously there is tumor heterogeneity. So cell-free DNA patients with metastatic breast cancer deserve to have that. And of course the allele frequency and the measure of response can be very frequently evaluated before imaging.
So the next question, do you sequence everybody at diagnosis?
I don't sequence everybody at diagnosis.
Did you not hear the last panel? Did you not hear Nelly and everybody else talking?
Yeah, I think we know.
You've got to hit early.
I think we should, certainly every tumor should be sequenced, and every individual should have a genetic testing so that we can actually really identify even other diseases, not only cancer in general. But it takes resources to implement that. You reduce the cost on one side and you increase on the other side. So the healthcare that is thinking about prevention, at least that part of the risk, the genetic risk, could be identified early. And of course, always the question, if you do NGS on the primary tumor, what you would do with the information, that we should answer that question.
Yeah. All right, thank you very much indeed, Massimo. Great. Raju, do you want to just sit there, or do you want to come up? You can sit if you want.
Everybody jokes about me about, whether I'm standing up or, people can't see me. But anyway, many of you probably don't know that Anna Barker was supposed to be on this panel. And unfortunately, Anna is not able to come. And Anna and I were going to talk about the role that the National Cancer Institute played in precision medicine. But who am I to talk about NCI? Here is the director of the NCI, and Doug is here, Dana is here, and but nevertheless, I'll say a little bit about, a little bit that I know. So in 2006, the National Cancer Institute decided on a pilot program to try to decipher the genetics and genomics of tumors. And Anna, together with the director of the NCI at that time, launched this program. And I had the privilege of being a part of that program. And the idea was to be able to get large numbers of tumors from patients who have not been treated before, and isolate nucleic acids from these tumors. Also get normal samples from all of these patients, and then use a variety of different genetic and genomic technologies to try to find out, what the constitutions of those tumors were. And during the period of time that the Cancer Genome Atlas was active, nearly 10,000 tumors have been examined, and with a variety of different technologies. And we learned a few lessons from examining those tumors. And I just tell you very briefly some of the highlights of them. So one of the things that was done for each of those tumors, all of the 20,000 genes in each of those tumors have been examined. And so one remarkable finding from that is that any given tumor type, there were only about 20 to 25 different genes that were ever found to be significantly mutated. So of the 20,000 genes in a given cancer, only a very small fraction of genes are important. The second important feature, that in a given tumor, there are only about a half a dozen or so different types of genetic and genomic changes. And that means that for the maintenance of that tumor, only those few genes are really responsible for them. The other thing that's actually very interesting that was found out is it turned out that all of these tumors that have been examined, a substantial portion of the tumors actually had germline mutations in oncogenes, tumor suppressor genes. And when those tumors were actually examined, it turned out that the same genes were, had 2 hits. And so actually, if you knew that all of those patients had those types of mutations and therefore were susceptible to be able to develop cancer, then we would have been able to do something, and prevention that our previous panels talked about. So, but one of the important things as a result of finding all of these mutations in tumors is that the way that we think about cancers really changed. As we all know that most of the cancers are defined by the tissue of origin, but when these mutations were found, that it is now possible to be able to define the tumors on the basis of the genetic compositions of the tumors. So what good is that? What is really interesting is what is good is that pharmaceutical companies have been developing drugs that target specific types of genetic changes that are present in these tumors, and therefore, if you can identify those patients that have particular types of genetic changes, that you would be able to identify the appropriate drug for which the patients will most likely be able to respond, and that's what precision medicine is in cancer. So that turns out to be a very important feature. And so if you look at all of the different, let us say, solid tumors, all of these 10,000, and you set up the total number of genes that are found to be mutated in all of those cancers, it actually turns out to be only about 500 or so. And many medical institutions as well as private commercial institutions have actually developed panels to test these 400 genes in any tumor type. And some institutions like our institution and many other institutions around the country now use those types of panels. So that any new patient that comes into the clinic, that they would be tested with these panels of 500 genes or so, and be able to define what the possible treatments that could be. And I think some of my colleagues here from Dana-Farber would be able to tell, and Monica probably knows about this too, is that now all of the physicians are so used to be able to getting this kind of information, for them to help make decisions, that if the reports are a day late, they're calling the path department and say, where is my report? And so it's really changing the practice of medicine and how you'd be able to treat the patients. So the question that Brian asked is that this is really fantastic. Is it being applied everywhere? Unfortunately, not everybody uses these types of tests that would really benefit the patients. And Doug talked about this a couple of years ago, about how you'd be able to use this information. And you talked about the idea, it said that you need to be able to show the effectiveness of these types of approaches. And if you could show that this testing and the subsequent treatments and the outcomes from those patients can be demonstrated, then that would be accepted universally. And that's what we're striving to, and that's where personalized or precision medicine today is, and there is a tremendous amount to try to push this idea of trying to being able to get all of these implemented across the country for all of the cancer patients and around the world.
Beautifully said. So can I just say, I asked a couple of people at my table just after Jim's talk and the first opening panel, and they were astounded at the progress over the last 25 years, and especially with the sequencing of the human genome and everything that Foundation Medicine and Tempus have been doing now for, I think it's 12, 13 years. Actually, it's 15. Why is this not more commonly adopted?
Well, as I pointed out, first of all, in the United States, as you know very well, the majority of the cancer patients are not treated at academic medical centers, but are treated in community oncology centers. And I had the opportunity to actually, in the last 10 years, visit a large numbers of those community oncology centers and so on and so forth. And of course, they want to be able to do the very best, for the patients. But the primary concerns are not necessarily about using these technologies. And as you know, Brian, I think changing the attitudes of people is very difficult to do. And that's one of the things that we talked about. Be able to show the effectiveness of using these kinds of technologies. And I think that's more and more of that is emerging now. We saw some of the examples, like today, as to what happens when you do these types of testing and treatments and whether those patients benefit. And there are really some remarkable results in many different cancers where there is a significant improvement of that. We need to be able to distribute that kind of information to everybody.
So do you have any idea what percentage of your patients in Harvard are being sequenced amongst the various academic?
At Dana-Farber, every new patient that comes in, their DNA tumor samples are subjected to treatment. Yeah.
That's what should be done. Just for your information, 85% of cancer patients are treated in the community. So it's only 15% who are benefiting from these fantastic academic centers. Any questions, any comments from the audience on this? We've just reached a point. Raju, do you know anything about sequencing internationally? Sequencing internationally.
Oh, actually, there are some people here. Hayan is here, and he might be able to, say something. And yes, I think there are countries, China that we know about, and Europe has made a decision that it will want to have regulations about how to test and interpret the information throughout the European Union, and so that's all taking off now. So there is a tremendous amount of movement.
Yeah, France is doing very well.
Yes.
As well. Yeah. Okay. Thank you very much. We'll move on. Min, will you take the platform, please?
My slides, please. Thank you, Brian. Thank you, Sujan, for inviting me to be here. It's my first time, along with Massimo. Both of us are newcomers. And from the color of the tie and shirt I'm wearing, what cancer type I'm working on. So this month is breast cancer. Next month is pancreatic cancer. So purple for pancreatic cancer. My name is Min Lee from University of Oklahoma Health Science Center. I'm the associate director for Stevenson Cancer Center, and received the NCI designation 5 years ago. And early this year, we were successfully renewed. Thanks for our director and the strong support from NCI. And we proudly serve the state of Oklahoma, which is a significantly understudied state, largely rural state. And my research focus is on pancreatic cancer, literally the worst cancer of all cancer types. Brain tumor, I know Al is going to talk about it. It's the second worst. And also work for a American Association for Pancreatology and APA, and which is the largest professional organization for pancreatic cancer, pancreatitis, and other pancreatic disease. And this year, the APA annual meeting is going to focus on metabolic reprogramming and cancer cachexia, which is another significantly underfunded, understudied disease. So next slide, please. So we all know that pancreatic cancer is the worst cancer of all cancer types. The overall survival, 5-year survival, is barely 12%. And I started my research career 20 years ago. It was 5%, and we didn't go that far. You look at that 5% to 12%, we did pretty good, but largely because we, able to identify pancreatic cancer in early stage. If you look at the stage 4, we were largely unchanged. There's not much has been done to improve the overall survival. The average survival time for end-stage pancreatic cancer is 3 to 6 months. So this is a very, very malignant disease. And the only effective treatment, a curative treatment for pancreatic cancer is a surgical procedure called Whipple procedure. But unfortunately, more than 85% are not eligible for surgery. They were diagnosed at late stage. We call the horses already out of the barn. There's not much we can do with it. And we talk about immunotherapy, we talk about virus therapy, especially oncolytic virus therapy, and small molecule inhibitors for years, especially targeting the KRAS non-targetable target. And you heard about talks earlier by Avery Posey from UPenn, that my dear colleague Courtney Houchin here as well. They did some outstanding work on developing the CAR T using CCAM6 and other people targeting mesothelin. And we also have some new targets in pancreatic cancer, zinc transporter, and so on. But still, there's a long way to go to develop a potent immunotherapy. I talked to Jim yesterday. Unfortunately, pancreatic cancer is a cold tumor. It's surrounded by fibrosis and a lot of tumor suppressive cells around that. So it's not a hot tumor, and we have tried a long way to try to boost the immune response against pancreatic cancer. But we still desperately need a novel therapeutic strategy and early target for pancreatic cancer. Next slide, please. So two bad things about pancreatic cancer. One is the lack of early detection and the highly resistant to traditional chemotherapy. The other one is also significantly understudied research area, what we call cancer cachexia. Most of the pancreatic cancer patients, they go through this wasting syndrome, called muscle wasting, and before that is the adipose loss, and which both of them lead to the rapid progress of pancreatic cancer and contribute to the, the overall survival. So about a decade ago, we started to look at this research area and how does pancreatic cancer develop cancer cachexia along with others, a couple of other cancers which is known for cachexia. One is lung cancer, the other one is colorectal cancer, maybe a little bit of head and neck cancer as well, but not much for brain tumor. So again, cancer cachexia is also a multiple organ disease. It's not a single disease. It involves the multiple interaction, crosstalk between tumor cells and immune cells, to name a few, macrophages and neutrophils, etc. It also involves the crosstalk and interaction between the tumor and the central nervous system. And of course, involves the target organs such as muscle and skeletal muscle, cardiac muscle, and adipocyte as well. So it's really a multi-organ syndrome that the signal comes from a tumor that sends out the cachexia factors and cachexia signals to the organ system that driving the progress of cancer cachexia, including muscle wasting and adipose, which is happening earlier. I don't think we have too much time to cover the molecular signaling that apparently there's a very complex signal transduction network involved in cancer cachexia. Just give you a few examples: TGF-beta pathway, R6-3 M-kappa B pathway, and the TWIC pathway, and also microRNA. Carlos here in the audience, the many microRNA was involved in cancer cachexia. So about 10 years ago, we started to look at how does a overexpress a marker. Just give you, for example, zinc transporter, which is driving the pancreatic cancer progress and metastasis, also drives the process of cancer cachexia, which involves the crosstalk between tumor cells and recruited the macrophage and causes a significant increase of a TWIC, which is known to be an inducer for cancer cachexia and muscle wasting. So I really hope through this conference we have the chance to raise awareness for pancreatic cancer and for cancer cachexia. And our director of NCI is here in the audience. I hope working with NFCR and we'll raise some awareness and raise more funding for this understudied research. Thank you so much.
Perfect. So George Sledge famously said about 12 years ago, we all know what's the origin of pancreatic cancer is, RAS. So we just don't have the drugs for it. What has been the progress in the RAS domain?
Well, Frank McCormick has been leading a RAS initiative, and we have been nonstop trying to develop inhibitors for KRAS. We made some progress, and apparently G12D is a big target. Lately, in the last few years, we have developed inhibitors for G12C and others. which benefits probably 1%, 2% pancreatic cancer patient, and we still have a long way to go. But KRAS is the biggest elephant in the room, but don't forget there's the other targets and other research areas that we really want to look at that. Just give you for examples, cancer cachexia and immune therapy. How do we improve the response to the immune therapy? I think that's the way to go as well. We talk about combinational therapy, and so yeah, KRAS is important, but that's not the only one that we're working on.
So you'll have new KRAS drugs, and you'll probably combine that with immunotherapy when you make the tumors hot.
Exactly.
Is that it?
Yeah.
All right, you've taught me something.
All right.
All right, great. Thank you so much.
Thank you.
My dear friend, you're next.
Right, next.
Yeah.
My name is Alfred Young. I'm from MD Anderson, but I've been working on brain tumor for many, many years. Now, take off on what Raju said. We want to talk about personalized medicine in brain tumor. We owe a lot of credit to Anna. I'm staying up here as a substitute for Anna Barker because she's sick. I want to wish her well, a rapid recovery, because I think Anna played a very major role in bringing personalized medicine to brain tumor, to glioblastoma, because Anna was the director of the TGCA program, and she selected glioblastoma as one of the 3 first tumors to be sequenced. And I was honored enough to be on the committee to work with Anna on the brain tumor project, on the glioblastoma project. The TGCA sequencing of 500, I think now we're more than 500 now, of glioblastoma, both newly diagnosed as well as recurrent. We placed a map, the molecular map, for us to understand the biology of glioblastoma. We also know a lot about it, but on the other hand, I must say, translating the knowledge of all the gene mutation that we discover into the clinic has been very difficult because of the heterogeneity of the tumor and evolution of the tumor cell from normal cell to tumor cell to low-grade tumor to high-grade tumor, and also post-treatment, really has placed a lot of challenge and barrier to bring the knowledge of all these molecular alteration that we know into the clinic to find the right medicine, right? So, however, though, the contribution of what we learned from TGCA actually bring the diagnosis and the classification of glioblastoma from the morphology basis to molecular basis. And then we need to thank Webb and David Lewis, the 2 work on bringing all these molecular knowledge into the clinic diagnosis and modify the way that we classify the tumor and also grouping them for not only prognostic indication as well as also for some therapeutic advantage. So the last 2 versions of the WHO, pathology book, they're edited by Webb and David Lewis, really plays a role of the change. Nowadays, as of 2021, the diagnosis of glioblastoma is no longer morphological based, it's molecular based. The classification and the groupings are based on how many genes are mutated, including especially IDH1, P53, and 1p19q co-deletion, and a couple of genes. But on the other hand, in terms of moving into therapy, the benefit that we get most from these molecular alteration is actually in pediatric tumor, a lower grade tumor, because now we learn IDH1 mutation separate the lower grade tumor to glioblastoma. Glioblastoma is only IDH wild type tumor. And all the tumors that had IDH mutation go to the lower grade tumor, whether it's astrocytoma or oligodendroglioma. The other alteration that's very critical for pediatric tumor and lower grade tumor is H3K20M, K27M mutation. That mutation allows us to understand the epigenetic change, epigenetic regulation of the midline and pontine glioma, which occur mostly in pediatric age group. And also allow us to really look into some medicine that will modify the epigenetic process. And that had therapeutic indication. The other gene that identified and influenced the pediatric tumor group and also bring it into the therapeutic range is the BRAF V600 mutation in low-grade glioma, optic glioma, and pilocytic astrocytoma in the pediatric age group. And because identified that mutation allows us a very high response rate of using MEK inhibitor. And so actually, FDA approved the use of trametinib and the dabrafenib for pediatric brain tumor patient with the BRAF V600 mutation. And that's a big. The other major, we just encountered a major benefit from molecular medicine is the IDH1 mutation and IDH1 inhibitor. And the latest studies show that using the IDH1 inhibitor allow us to prolong and delay the recurrence or the progression of low-grade glioma to high-grade glioma. And actually allow us to postpone or push back the use of radiation in the pediatric age group, and which really greatly improve the quality of life for those patients. The other benefit, I think I must end with another major contribution of Anna and Webb in terms of applying precision medicine to glioblastoma. In 2013, Webb, Anna, and I sit together and say, well, we need to do something about glioblastoma. What we have been doing does not work. And with the finding of the TGCA, we now need to really find a way to test drug quickly because of the heterogeneity of the tumor. We don't have a dominant gene, but we have many things that we need to do to test them, and we need to test them quickly in the patient. And so after 2 years of deliberation with a bunch of clinicians, scientists, with the help of NFCR and AFCR, and we decided that we need to create a platform to do a adapted design platform trial, allow us to test multiple drugs at the same time. And that born the GBM Agile platform trial. And we also thank to the NCI and FDA, it actually allow us to create that platform trial to seamlessly go from phase 2 to phase 3. If we have a positive finding, we can get a drug registered with fewer patients. And so with that platform that and Webb and work on now, we're able to really cut down the number of patients that needed to test the drug from phase 2 to phase 3, and then they get drugs approved and allow us to test multiple drugs at the same time. Right now, the protocol is open, and we are testing 4 drugs at the same time right now. So I'm going to end up here. Yeah.
So Al, can I just ask you, beautiful, can I just ask you about time frame? So when I was junior faculty at Sloan Kettering, we were given 2 diseases, one nice lymphoma and one terrible, which was breast. Now, breast cancer is 85% cured. So what is the time frame, do you think, in glioma? Is it going to be major advances in 10 years? Less?
I think glioblastoma is a tough nut to crack. I always say, when I started at MD Anderson in 1981, I modeled my learning to non-small cell lung cancer. I tried to use every drug that that they use in non-small cell lung cancer. Because at that time, heterogeneity issue is the same with non-small cell and glioblastoma. Drug that we use are kind of the same, platinum, and so I always try to learn from non-small cell lung cancer. But look at how far non-small cell lung cancer has come up in terms of from precision medicines, and we're still down here. Now, the problem we have is heterogeneity of the tumor, intra-tumor heterogeneity, inter-tumor heterogeneity, and cellular heterogeneity, as well as the protection of the brain by the blood-brain barrier, not allowing drugs to go in. And that really hampers, in terms of how far we go. And the lack of driver gene, we have to deal with multiple genes and also multiple pathway that kind of overlap with each other and provide an escape pathway. That also really, I don't know how long it will take us to really crack this nut. And I'm not going to say that we can cure the disease in 10 years. I think we are doing better in terms of improving the median survival. We are also doing better in raising, just like Jim said, raising the long-term survival rate. But we're not close. I don't think we're not close. We need a lot of, and the other thing that I also make an appeal in terms of the timeline is we need more talent to come into the brain tumor field, especially glioblastoma. And we have a lot of talent, top scientists working in lung cancer, breast cancer, even pancreatic cancer. We don't have enough top-flight scientists and clinician scientists working glioblastoma, and we need them.
Thank you. Can I just ask the audience, I mean, you have one of the top, if not the best neuro-oncologist in the world in front of you. Do you have any questions? I know. All right, we'll let you go, my friend. Drew, come on up.
Is it okay if I just
Yep.
sit here?
Yep.
Okay, can folks hear me okay? Okay. So when Haley called me a couple days ago to ask me to pinch it, having just been through our Institute's EAB meeting, I said, I'm happy to do it as long as I don't have to put together another slide deck. She said, done deal. So in my world of cancer immunotherapy, biomarkers and precision medicine are somewhat different than conventionally for oncogene-targeted therapies, in that for an oncogene-targeted therapy, a biomarker is relatively black and white, in the sense that if, as you heard from Dr. Young and with melanoma, if you do not have a BRAF mutation, so if you're in that 55% of melanomas that doesn't have a BRAF mutation, you will not respond to a BRAF inhibitor. If you're an earlier stage glioma, if you do not have an IDH1 mutation, do not bother trying an IDH1 inhibitor. In the world of immunotherapy, with one wonderful exception, which is the mismatch repair deficiencies, which really translate to a high mutational load, which translates to high antigenicity for that tumor, lots of antigens that the patient's T cells can recognize. But with that as in a sense, an exception, it's really the expression of molecules that regulate immune responses and their levels of expression that represent the biomarkers. Still the dominant biomarker for identification of patients that will have a higher probability of responding to anti-PD-1 or anti-PD-L1, as shown by Dr. Topalian, together with with Janice Taub, our lead immune pathologist, is expression levels of PD-L1. Those biomarkers tend to be dirtier than specific mutations, but nonetheless, when one thinks about the landscape of molecules that we're learning about and that are targets for a vast array of antibodies and other engineered immunotherapies, including engineered T cells, this really offers an opportunity. I think we've tapped clinically about 0.0001%. And so I want to just throw out a couple of challenges. So if you look at the fact that we now at the single cell level can analyze the expression level of every gene in the genome, we are now moving toward the ability to spatially profile every gene in the genome. One can now envision, Alana Fertig, who's here, her lab is actually down the hall from ours, that you can take this unbelievable massive information and be able to predict extremely effectively who is going to respond not only to one drug or the other drug, but to really combinations of drugs. Again, in the immunotherapy world, there are literally thousands of combination trials, and immunotherapy is no longer just an antibody against a checkpoint or a vaccine. I remember Jim saying every therapy is an immunotherapy, and that's true. Standard of care for most non-small cell lung cancer patients, if their PD-L1 levels are below 50%, if it's above, it's only an anti-PD-1. If it's below, it's anti-PD-1 plus chemotherapy. So challenge, but yet you now look at how this is done, and all of the immunohistochemistry that we do in standard of care, it's still single marker immunohistochemistry. There is not yet a single standard of care that looks at the co-expression of two different molecules. Our pathology colleague Janice Taub got together a number of years ago with an astronomer at Hopkins, who is one of the world's leaders in development of, Alex Saleh is his name, in development of algorithms to analyze astronomical images. They got together and he applied that to immunofluorescence, and with only 5 or 6 markers, now they call it astropath, they were able to define for the first time high risk versus intermediate versus low probability responders in melanoma to anti-PD-1. So that's challenge one. How do we take this ever-expanding technology, both technical as well as computational, and translate that into something real world? Because every cancer patient gets a biopsy and it gets plunked into formalin and it gets put into paraffin. So there is so much information that you have there. Also, every cancer patient gets their blood drawn. So you can do ctDNA. That combination should be able to give us amazing precision. But if you actually look what is standard of care now, it still has not changed that much from literally 40 or 50 years ago. when Elaine Jaffe was introducing literally the first application of monoclonal antibodies clinically. So that's number one. The second challenge is uptake. So outside of colon cancer, it is still a minority of patients in the United States that get tested for mismatch repair deficiency mutations. Why is this not done as routinely as hematocrit? Because if you have a mismatch repair defective tumor, then anti-PD-1 should be part of your regimen. You shouldn't even, it should be a no-brainer. I get calls from folks with endometrial cancer. A third of endometrial cancers are mismatch repair defective. It is still way, way less than half of patients with endometrial cancers that actually get tested for MMRD. So that's number one. The second has to do with screening. So I had the opportunity to be involved with Suzanne in in really the earliest studies testing neoadjuvant application of anti-PD-1 in lung cancer. This was led by Patrick Ford. That translated very quickly to an FDA approval of neoadjuvant anti-PD-1 and plus chemotherapy in stage 2 and 3 lung cancer. That was actually the second FDA approval. The first, thanks to the I-SPY format, was in breast cancer for high-risk breast cancer neoadjuvant combination anti-PD-1 and chemotherapy. Looking at the lung cancer, now low-dose CT scans are approved for individuals that are at high risk of lung cancer. Namely, the best biomarker of all is, do you have a smoking history? If you look at the improvement in 2-year relapse-free survival with neoadjuvant PD-1 chemotherapy in stage 2 and 3, that's saving potentially 20% of the lives of these lung cancer patients.
Drew, can I beg you to bring it soon to a close?
Yes, so I'm going to say,
flashing all around me.
right, so one number, the rate of screening for lung cancer in high-risk individuals in the state of Maryland is 5%. So we are losing 95% of individuals just because only 5% get screened, that of which probably 20% could be cured who otherwise wouldn't be. So that's the second challenge. How is this information, which we know, which is available as standard of care, how are we going to get this out to the public?
Drew, thank you. Thank you so much. I want to thank my superb panel. They're all brilliant. I wish to thank the audience. I think you've had 5 beautiful dissertations today. I hope you've learned something. And also, I do want to thank NCI. Monica, I spent 7 wonderful years there myself, trained, so many initiatives which you've led forward and which your past directors have led forward. So thank you. Continue pouring the money out, please. God bless.